Q1. What is the mechanism of action of the Clostridium botulinum toxin? 1. Blocks GABA receptors in motor neurons. 2. Inhibits acetylcholine release at the neuromuscular junction. 3. Activates acetylcholinesterase. 4. Stimulates muscle sodium channels. Answer 2. Inhibits acetylcholine release at the neuromuscular junction. Botulinum toxin blocks presynaptic ACH release, causing flaccid paralysis. Q2. Which of the following bacteria is catalase positive and commonly causes food poisoning via enterotoxin production? 1. Streptococcus pyogenes. 2. Clostridium perfringens. 3. Staphylococcus aureus. 4. Streptococcus pneumonia. Answer: 3. Staphylococcus aureus. It produces heat-stable enterotoxins and is catalase positive, causing toxin-type food poisoning. Q3. Which gram-positive rod forms spores and causes pseudomembranous colitis after broad-spectrum antibiotic use? 1. Corynebacterium diphtheriae. 2. Clostridium difficile. 3. Bacillus anthracis. 4. Streptomyces griseus. 2. Clostridium difficile. It overgrows due to antibiotic-induced dysbiosis and produces enterotoxins leading to colitis. Q4. Which virulence factor allows Streptococcus pyogenes to evade phagocytosis by binding complement C3? 1. Lipoteichoic acid. 2. Streptolysin S. 3. M protein. 4. Hyaluronidase. Answer: 3. M protein. M protein binds C3 and prevents opsonization, allowing immune evasion. Q5. Which of the following is a spore-forming anaerobic bacillus that produces alpha toxin causing gas gangrene? 1. Clostridium tetani. 2. Clostridium botulinum. 3. Clostridium perfringens. 4. Mycobacterium lep. Answer: 3. Clostridium perfringens. Alpha toxin causes tissue necrosis and gas production in muscles. Q6. Which organism causes diphtheria and produces a toxin that irreversibly inhibits EF2, blocking protein synthesis? 1. Corynebacterium diphtheria. 2. Mycoplasma pneumoniae. 3. Bacillus cereus. 4. Propionibacterium acnes. Answer: 1. Corynebacterium diphtheria. The diphtheria toxin binds EF2, halting translation and leading to tissue necrosis. Q7. Which of the following bacteria lacks a cell wall and requires cholesterol for membrane stability? 1. Mycobacterium tuberculosis. 2. Streptococcus agalactiae. 3. Mycoplasma pneumoniae. 4. Actinomyces israelii. Answer: 3. Mycoplasma pneumoniae. It is wall-less and incorporates host cholesterol in its cell membrane. Q8. Which gram-positive cocci is beta-hemolytic and associated with scarlet fever due to erythrogenic toxin? 1. Staphylococcus aureus. 2. Streptococcus pneumonia. 3. Streptococcus pyogenes. 4. Enterococcus faecalis. Answer: Streptococcus pyogenes. Group A. Streptococcus produces erythrogenic toxin, leading to scarlet fever. Q9. Which component of Bacillus anthracis facilitates host cell entry of edema and lethal factors? 1. M protein. 2. Protective antigen. 3. Poly-D-glutamate capsule. 4. Anthrolysin. Answer: 2. Protective antigen PA binds host cells and enables entry of EF and LF to form anthrax toxin complexes. Q10. Which of the following agents is part of the standard first-line therapy for tuberculosis? 1. Clarithromycin. 2. Metronidazole. 3. Isoniazid. 4. Amoxicillin. Answer: 3. Isoniazid. INH inhibits mycolic acid synthesis in Mycobacterium tuberculosis. Let me know if you'd like. Q11. Which of the following best describes the gram-positive coccus that produces TSST1 and causes toxic shock syndrome? 1. Staphylococcus epidermidis. 2. Streptococcus pneumonia. 3. Streptococcus agalaxia. 4. Staphylococcus aureus. Answer: 4. Staphylococcus aureus. It secretes toxic shock syndrome toxin 1, TSST1, a superantigen causing systemic inflammation. Q12. Which diagnostic test is used to confirm infection with Streptococcus agalaxia, group B Streptococcus? 1. Coagulase test. 2. CAMP test. 3. Dick test. 4. Schick test. Answer: CAMP test. The CAMP test shows enhanced hemolysis when GBS is cultured with S. aureus. Q13. What is the primary antibiotic treatment for Streptococcus pneumoniae infections? 1. Vancomycin. 2. Doxycycline. 3. Penicillin. 4. Rifampin. Answer: penicillin. S. pneumoniae is generally sensitive to penicillin, though resistance is rising. Q14. Which toxin produced by Clostridium tetani inhibits the release of inhibitory neurotransmitters? 1. Tetanolysin. 2. Botulinum toxin. 3. Tetanospasmin. 4. Alpha toxin. Answer 3. Tetanospasmin. Tetanospasmin prevents GABA and glycine release, leading to spastic paralysis. Q15. Which component of Streptococcus pyogenes impairs phagocytosis by mimicking host connective tissue? 1. Lipoteichoic acid. 2. C5a peptidase. 3. Streptolysin O. 4. Hyaluronic acid capsule. Answer 4. Hyaluronic acid capsule, it mimics host connective tissue and evades immune detection. 16. Which treatment is most appropriate for a Clostridium difficile infection? 1. Intravenous ceftriaxone. 2. Oral vancomycin. 3. Topical clindamycin. 4. Subcutaneous gentamicin. Answer 2. Oral vancomycin. Oral vancomycin is effective for localized infection in the GI tract. Q17. Bacillus anthracis forms a capsule composed of which of the following? 1. Hyaluronic acid. 2. Polypeptide D-glutamic acid. 3. Polysaccharide. 4. Mycolic acid. Answer 2. Polypeptide D-glutamic acid. Unique among capsules, it's protein-based rather than polysaccharide-based. Q18. Which of the following bacterial species is acid-fast due to the presence of mycolic acid in its cell wall? 1. Corynebacterium diphtheria. 2. Bacillus anthracis. 3. Mycobacterium tuberculosis. 4. Streptococcus agalactiae. Answer 3. Mycobacterium tuberculosis. Mycolic acids in the cell wall make it acid-fast and resistant to standard staining. Q19. Which test is used to assess immunity or exposure to diphtheria toxin? 1. Mantoux test. 2. Camp test. 3. Dick test. 4. Schick test. Answer 4. Schick test. The Schick test assesses susceptibility to diphtheria by intradermal toxin injection. Which bacterial pathogen causes nodular white colonies resembling molar teeth and is associated with oral abscesses? 1. Actinomyces israelii. 2. Propionibacterium acnes. 3. Streptomyces griseus. 4. Bifidobacterium bifidum. Answer 1. Actinomyces israelii. It is known for forming sulfur granules and molar tooth-like colonies in anaerobic culture. Let me know if Q21. Which gram-positive bacillus is responsible for causing acne and is part of the normal skin flora? 1. Bifidobacterium bifidum. 2. Actinomyces israelii. 3. Propionibacterium acnes. 4. Mycobacterium leprae. Answer 3. Propionibacterium acnes. It resides in sebaceous follicles and contributes to acne pathogenesis. Q22. Which bacterial genus is known for producing the highest number of clinically useful antibiotics? 1. Bacillus, Streptomyces. 3. Corynebacterium. 4. Clostridium. Answer 2. Streptomyces. Streptomyces species produce antibiotics like streptomycin and tetracycline. Q23. Which gram-positive bacillus forms pseudomembranes in the pharynx and can lead to respiratory obstruction? 1. Mycobacterium tuberculosis. 2. Bacillus cereus. 3. Corynebacterium diphtheria. 4. Streptococcus pyogenes. Answer 3. Corynebacterium diphtheria. The diphtheria toxin can induce pseudomembrane formation and suffocation. Q24. Which organism causes leprosy and is known for its inability to be cultured in artificial media? 1. Mycobacterium avium. 2. Mycobacterium leprae. 3. Corynebacterium jicaeum. 4. Mycoplasma pneumoniae. Answer 2. Mycobacterium leprae. It cannot be grown in vitro and has the longest doubling time of human pathogens. Q25. Which of the following gram-positive rods is used as a probiotic and is found in the gut microbiome? 1. Bifidobacterium bifidum. 2. Propionibacterium acnes. 3. Clostridium perfringens. 4. Mycobacterium leprae. Answer 1. Bifidobacterium bifidum. It's a lactic acid-producing bacterium important in gut flora and fermented products. Q26. Which organism has no cell wall and is resistant to beta-lactam antibiotics? 1. Corynebacterium diphtheria. 2. Streptococcus agalactiae. 3. Mycoplasma pneumoniae. 4. Mycobacterium tuberculosis. Answer 3. Mycoplasma pneumoniae. It lacks a peptidoglycan cell wall, rendering penicillins ineffective. Q27. Which gram-positive bacterium causes neonatal meningitis and septicemia and is identified by a positive CAMP test? 1. Staphylococcus epidermidis. 2. Streptococcus agalactiae. 3. Streptococcus pneumonia. 4. Listeria monocytogenes. Answer 2. Streptococcus agalactiae, group B streptococcus, causes serious perinatal infections in neonates. Q28. Which organism causes gas gangrene by producing a lecithinase, alpha toxin? 1. Clostridium tani. 2. Clostridium botulinum. 3. Clostridium perfringens. 4. Clostridium difficile. Answer: Clostridium perfringens. Alpha toxin destroys phospholipids in membranes, causing tissue necrosis and gas formation. Q29. Which gram-positive coccus is ahemolytic and exhibits optochin sensitivity on blood agar? 1. Staphylococcus saprophyticus. 2. Streptococcus mitis. 3. Streptococcus pneumoniae. 4. Enterococcus faecalis. Answer: 3. Streptococcus pneumoniae. It shows ahemolysis and is optochin sensitive, distinguishing it from other viridans streptococci. Q30. Which of the following bacteria is associated with oral sulfur granules and molar tooth colony morphology? 1. Streptomyces colicolor. 2. Actinomyces israelii. 3. Nocardia asteroides. 4. Bifidobacterium breve. Answer: 2. Actinomyces israelii. It forms branching filaments and yellow sulfur granules in anaerobic infections. Let me know. Q41. Which gram-positive bacterium possesses protein A that binds the Fc region of IgG and inhibits opsonization? 1. Staphylococcus saprophyticus. 2. Enterococcus faecium. 3. Staphylococcus aureus. 4. Micrococcus luteus. Answer: 3. Staphylococcus aureus. Protein A interferes with immune clearance by binding the Fc portion of IgG antibodies. Q42. Which component of bacillus anthracis is responsible for edema by disrupting water ion homeostasis in host cells? 1. Protective antigen. 2. Lethal factor. 3. Edema factor. 4. Alpha toxin. Answer: 3. Edema factor. EF increases intracellular CAMP, leading to edema and immune evasion. Q43. What is the main virulence factor of Clostridium perfringens responsible for gas gangrene and tissue necrosis? 1. Lipid A. 2. Alpha toxin. 3. Streptolysin S. 4. Lecithinase B. Answer: 2. Alpha toxin, a phospholipase C that destroys host cell membranes and promotes necrosis. Q44. Which diagnostic test is used to determine susceptibility to diphtheria toxin? 1. Schick test. 2. Dick test. 3. Tuberculin skin test. 4. Camp test. Answer: 1. Schick test. This skin test determines whether an individual has protective immunity to diphtheria. Q45. Which gram-positive coccobacillus is motile, facultative intracellular, and can cross the placenta to infect the fetus? 1. Listeria monocytogenes. 2. Streptococcus agalactiae. 3. Clostridium difficile. 4. Corynebacterium pseudotuberculosis. Answer: Listeria monocytogenes, known for causing meningitis and sepsis in neonates via vertical transmission. Q46. Which bacterial genus is the smallest free-living organism, lacks a cell wall, and contains sterols in its membrane? 1. Mycobacterium. 2. Streptococcus. 3. Mycoplasma. 4. Actinomyces. Answer: Mycoplasma. It survives without a cell wall by incorporating cholesterol into its membrane. Q47. Which acid-fast bacterium has the longest generation time and cannot be cultured in artificial media? 1. Mycobacterium kansasi. 2. Mycobacterium avium. 3. Mycobacterium leprae. 4. Mycobacterium fortuitum. Answer: 3. Mycobacterium leprae. It has a very long incubation period and only grows in animal models like armadillos. Q48. Which test uses purified protein derivative, PPD, to detect prior exposure to Mycobacterium tuberculosis? 1. Mantoux test. 2. Schick test. 3. Camp test. 4. Weidel test. Answer: 1. Mantoux test. This intradermal test evaluates delayed type hypersensitivity to M. tuberculosis antigens. Q49. Which substance accumulates in Corynebacterium diphtheria and appears as polar granules on Neisser stain? 1. Lipid A. 2. Mycolic acid. 3. Polyphosphate volutin. 4. Capsule polysaccharide. Answer: 3. Polyphosphate volutin appears black on Neisser stain. Represents metachromatic granules important for diagnosis. Q50. Which gram-positive bacillus has a polypeptide capsule composed of D-glutamic acid instead of a polysaccharide? 1. Clostridium tetani. 2. Mycobacterium tuberculosis. 3. Corynebacterium diphtheria. 4. Bacillus anthracis. Answer: for Bacillus anthracis, unique for its D-glutamate capsule that contributes to its virulence and resistance to phagocytosis. Let me know